We're going to work through genetic and inheritance again because it's an important concept, and I know we started it at the end of term 1 and we worked through some items, but I just think it's better if we continue and we have a recorded session of this. So remember, parents always produce offspring that look like them, but they may differ in some ways. Genetics, which is the science of heredity, explains how these characteristics are passed on from one generation to the next. In this topic, we're going to explore some important concepts in genetics. These concepts, which are listed here, this is topic 5 that you're working through from your textbook, explain the inheritance of different characteristics. They also include the inheritance of genetic diseases. We will then look at the work of geneticists, which includes trying to replace damaged or missed genes that can result in a genetic disease. Producing products that are needed by people with these diseases and improving crops for better production. So this all boils down to how we can actually improve our lives by understanding the genetic concept. Our unit 1 is genes and genetic concepts. So when we look at genes, we need to understand that it is a section of DNA in a chromosome which carries a code about a particular characteristic of an organism. Example, your hair color, right? Everybody's got a different hair color, and that's specifically coded for in our DNA. So here at the bottom, here we've got our gene segment. This gene segment over here is a very important concept to understand how DNA and heredity comes into play. Now, scientists or geneticists think that there are about 25,000 genes on the chromosome in each human cell. So let's talk about this chromosome quickly. Let's get a blue pen out. Now, this chromosome is found, you should already know this, in the nucleolus, right? Or the nucleus. The nucleus is the brain box of a cell. And this nucleus has all our chromosomes. We know that we have 46 chromatids, or we've got 23 pairs of chromosomes that we end up dealing with in a human karyotype. So we have this in our nucleus, and in the nucleus we've got our chromosome. Now this chromosome has got DNA on it. It's got DNA in it, and because it's got DNA in it, the DNA codes for a specific gene, right? So there we've got a specific space. Let's say that space is coding for hair color. It means that its friend or its buddy on the dad's chromosome DNA, because remember you get one from mom and one from dad, the same space on the dad's chromosome will also code for hair color. So dad might be blonde and mom might be brown, and then you end up getting both of those or either of them depending on how meiosis has segregated these chromosomes, and we'll talk more about that a little bit later. But we know that this chromosome has the genes in that specific location. When you're talking about a gene in a location, we speak about the word locus, not like grasshoppers that come and attack you. It's a locus. A locus is the position of a gene on a chromosome. So it's the position of the gene on a chromosome. Now remember, you guys studied that in your somatic cells you have diploid chromosomes, hence the reason why you're going to have a mom and a dad. This is your diploid storage station, right? We keep all our DNA in one place. It's our 2N. And we know that half of that comes from mom and the other half comes from dad. And that's the one N and the other N, and they merge together because of sperm and egg, and then you end up getting your two ends. Okay. These homologous pairs are identical. The mom's pair and the dad's pair is identical to each other. The only thing that is not identical is dad has an X and a Y, and mom has an X and an X on a specific chromosome. And you guys know what these ones are called. These are called your sex chromosomes. I'm not going to have space to write it in there. But there you got your sex chromosomes. Okay. Guys, how does this actually code? Well, we know that this chromosome is wound up into a chromatin network, and the way it's wound up is by taking your double helix strand, wrapping it around these things called histones, and these histones together with the wrapped up DNA is called a nucleosome. And that nucleosome unraveled and unraveled and unraveled and unraveled gives us our genetic coding down here. And we already know that A has to pair up with T, that T has to pair up with A. And then what ends up happening is we need to code for proteins. This whole process from translation, if you can't remember all of these things, you need to go look at your notes. Translation and transcription allows us to create proteins. We call this protein synthesis, right? Put an I in there. So we create proteins. Proteins is actually what builds us up into who we are. So all the items that we are made up of was read from a gene segment. Okay, so genes is a section of DNA in a chromosome which carries a code about a particular characteristic. This characteristic, wherever that code is found on your chromosome, the location of that code is called your locus. And remember, your locus will be the same for mom and dad. The only time it will not be the same is because we end up having our sex chromosomes having something different. Dad has a Y, mom doesn't have a Y. So those are not going to be identical. Okay, but otherwise, every other chromosome pair, every other homologous chromosome will end up having the same location dictating the same code. And that's why genetics and inheritance is so important to understand. We're going to look at dominant and recessive genes in this section, but what we want to talk about first is how mutations allowed for these dominant and recessive genes to exist. Now, mutations have come about throughout the evolution of an organism. A mutation can be successful or it can be unsuccessful, right? A lot of people have a negative connotation towards mutation where they think that a mutation always leads to cancer. And yeah, cancer is a mutation of the cell that ends up growing and growing without stopping. So that is a problem. But another thing that a mutation could do is it could allow you to survive better than your peers. For instance, let's take a very random example. Whether this happens or not, I don't know. But if we look at a chameleon has the ability to change its skin tone. But what if back in the day, before chameleons were just little lizard-type organisms or whatever they are, and they would sit on green parts of the tree? And then one day, there was a slight mutation in its skin color that allowed it, when it moved to different parts of the tree, it could now change its skin color. So we've got our chameleon, and it's now climbing on trees. It used to only be green, but now a small mutation in its skin tone, in its skin, allows it to change its skin tone. And this change in its skin tone is actually quite a successful thing because it now doesn't only have to sit on the green leafy parts, it can go move on to bark, it can go move on to anything, and be camouflaged. So when you're thinking about mutation, don't always think about it as a negative item. Okay, now this mutation is specifically looking at a small change in the part of a DNA. And because of that small change, the instructions it codes for will also then change. Think about it. I have a code in this DNA, and that code is MAT. You and I know what a MAT is. We know what it looks like. We know how it feels. We know what it does, right? But now what happens if I go and change just one part of that sentence, and I change it with an S? Now it becomes SAT. It's something completely different to what it had. This is a hectic change, like this is changing it from one item to a completely other item, but the gist is still the same. If you're changing the code here, it's going to code for something different. Any small change in the parts of a DNA is actually quite a good thing sometimes. It allows for you to survive better. So these small mutations will allow for several different alleles. Now, what is an allele? An allele is just different variations. Oh, sorry, it keeps doing that. Different variations of the same gene. Now, let's look at it. We have hair color. We code for hair color dark brown as a big B, right? But we could also have, so let's call that brown, right? We code for it with a big B. But we could also have blonde hair. And we can code for that as a smaller b, okay? These are your alleles. This is what dictates what you're going to have. It's your phenotype. It's your genotype that dictates your phenotype. And we'll talk about that a little bit later, but come back and watch this video again when you understand that terminology. So when there are two alleles for the same gene, we call them multiple alleles. Now, hair color has brown hair, it has blonde hair, you have red hair, you have strawberry pink hair. These are all blends of these alleles bonding together. But the brown and the blonde is standard, so that will code for only those two types of hairs. We know that somatic cells will only have two alleles for each gene. Why do we know that? Because our chromosomes, our homologous pair, one is from mom and one is from dad, and each one has a specific section on it. Let's call it that section over there. Sorry. That section over there and that section over there. That could possibly code for your hair color. And mom could be brown haired and dad could be blonde haired. And when these two blend together, they give you whatever hair color it is that you've got. So somatic cells, which we know are our body cells, will only have two alleles for each gene. They can't have, so now we've got another allele for red. They can't have red as well as brown as well as blonde. They can only have two of them. Okay. One will generally be a dominant and the other one will be recessive, but it's not always like that. Sometimes we'll have a blend of these items together. But the dominant allele, dominant means it outweighs everything else, right? Dominant determines the phenotype. Your phenotype is your physical, your physical characteristic. So if I have, for instance, capital B, one from mom, one from dad. I have two B's, I'm going to have brown hair as a child. But let's talk about it. What happens if I have a little b and a little b? That means I have one from mom and dad, so I'm going to have blonde hair. But now let's, for instance, say I got one from dad and the other one from mom. Got a capital B and a small b. This is my dominant allele, and that is going to dictate the fact that I'm going to mask the blonde, and I'm not going to see that. I do still have it in my genetic makeup, so I can pass on that blonde to my child, but I can pass on this brown to my child, because remember, meiosis splits these items up. But in this case, the big B, which is your brown genotype, has now masked your little b, which is your blonde phenotype. And as a result of that, you as an individual presents, which is your phenotype, you present as having brown hair. Your recessive alleles, that's your physical appearance, will only determine the phenotype if no dominant allele exists. In other words, if I've got a recessive allele, which is the small b and the small b, if the dominant allele is not in this, then we'll only be able to present ourselves as being blonde. Blonde. Okay, but if I have a capital B and a capital B, my genotypes from mom and dad are two capitals, I'm going to present as brown. If I have a capital and a recessive, or a dominant and a recessive, I'm going to then present as brown. The only time my recessive allele will show its physical appearance is if it has no dominant alleles existing in it. Okay. Gregor Mendel was the father of our genetics. Sorry about that spelling mistake up there. That's genetics. Put an E in between there. Okay. Go watch this little video on him. It's quite an interesting just introduction to who he is and how it contributed to how we understand genetics today. But what he ended up doing is he ended up taking pea plants and he crossed them to see what the offspring ended up being like. But he did it in such a way that he was quite specific about using purebred plants. Now, what's a purebred plant? It's a plant that when it self-pollinates, so when self-pollination occurs, self-pollination, in other words, it pollinates with its own kind, itself, right? When it pollinates, self-pollination occurs, the offspring will always look like the parent plant. So he went and took contrasting traits, for example, a tall plant. So here's a nice little tall plant. And he took a short plant, right? And he would cross these together and he would see the offspring. And what he ended up realizing is these offspring all look the same. Okay, so now we have the offspring. But once he let the offspring self-pollinate, so whatever the offspring was, maybe it was just a medium-sized plant, not a large or a small. It was a medium-sized plant. But when he put two medium, well, not two medium plants, when he self-pollinated it, which means this plant has a male flower, let's do the male flower, and we have the female flower. When these two pollinated itself, when it self-pollinated itself, it ended up bringing out a certain ratio of characteristics. And depending on the type of cross that ended up happening, that ratio changed. And we're going to look at those ratios and we're going to understand them and we're going to predict them from specific crosses that we end up doing. Okay, so he then suggested that there must be something in the cells. So he realized that each of these plants end up having cells, right? And within these cells, we know now that there's these things called chromosomes. And these chromosomes carry out genes. And these genes code for life. And what we ended up realizing is that these genes, he didn't have a word for it at that point, but he called them factors. These specific factors in each cell will control the specific characteristics like height or color. He published his work in 1866, but scientists did not understand his findings until some time after his death, when the chromosomes were discovered, and they began to think that each chromosome might actually carry these factors. And Mendel was then referred to, and they then called these genes factor genes. Okay, so he used purebreds, he created offspring, he then allowed those offspring to self-pollinate, and specific ratios of characteristics came out there. We're going to do crosses to be able to identify those specific ratios.